Hello everyone, hope you guys are doing great. Myself Sheikh Fidos. Today we're going to be see the phase contrast microscopy principle. So first, let me see the microscope introduction in small. The microscope word can be split into the two separate words, that is a micro and the scope. The micro means a small or tiny and scope to view. The microscope can be classified as follows depending on the number of lenses and depending on the number of the eyepieces and depending on the source. Depending on the number of lenses, there is a simple microscope and compound microscope. It's described in a two microscope in this number of lenses. The means the single lens and the two lenses. In single lens, that means the example is given in this picture. This is a one lenses and the two lenses here. And depending on the number of eyepieces, that is monocular eye microscope and binocular microscope. That means this is a eyepiece. This is eyepiece from where we can observe our specimen. And depending on this, there is a monocular microscope and this one is binocular. And depending on the sources, light or optical microscope and electron microscope. Light or optical, here the optical lens is used and here the electromagnetic lens is used and here the visible light is used and here the electron beam is used. Now in microscope, we see the light in optical microscope in previous slide. So here it divided into bright field microscope, dark field microscope, fluorescence and phase contrast microscope. In bright field microscope, the specimen will be a dark image against a brighter background. Here you can see this is a dark image and the background is brighter. This is the brighter background. And here in dark field microscope produces a bright image. Here you can see the image, this image is bright and the background is dark. In fluorescence, the optical microscope that uses a fluorescence, and in phase contrast microscope we are going to see this in detail. In electron microscope, transmission electron microscope and scanning electron microscope. Now we'll see phase contrast microscopy. Now, why we are using a phase contrast microscopy if we have the dark field microscopy and bright field microscopy? If you know, the specimen should be stained if you are using the bright field microscopy or dark field microscopy. So in this phase contrast microscopy, the specimen should not be stained. It's not a compulsory. If you see the transparent cells or a transparent microorganism, the stain will going to be toxic for it. It will going to be die and we can't see the proper image in that. So here we are going to use a phase contrast microscopy for that type of a cells. That is, unstained living cell absorb a particularly no light. A poor light absorption result in extremely small differences in intensity distribution in the image. This makes the cell barely or not at all visible in bright field microscope. That's why we are using this phase contrast microscope. And it is first described or first invented by the Dutch scientist. That is, his name is Frits Wernicke. Now we'll see the principle of the phase contrast microscopy. Now this is a light. You can see this is an annular diaphragm. This is this shaded one will not allow the light to pass from it. The ring, you can see this white ring will going to allow the light to pass from it. And this condenser present over here, this condenser will going to condense this light on the specimen. This is a specimen, and this specimen, from this specimen the light will going to be passed through the objective lens here, and here there is this is unretarded rays and this one is the retarded rays. Why this is a retarded and this unretarded? If you see, this is specimen and the light rays, that means this refractive index, this refractive index passes, and when it hits to the organelle of our slide, if it hits to it, then the refractive index will going to be changed, will going to be small. Why? Because if you know this, the distance between these two peaks is lambda. So here the lambda is less. Therefore the objective or the optical length, optical density will less. That's why it's decreasing. And this retarded and this unretarded light will going to be go through the phase split. Why? Because this refractive index Ri, we can't able to see it with our eye. We can see only intensity. That means dim light, bright light, our eye it can catch that, not this refractive index. So here the scientist placed this phase plate, and under this phase plate there is a phase ring, due to which this light will going to be converted, and the image plane will going to be form of the microorganism. We can see the microorganism over here with our eye. It will going to convert in that way that we can see, the human eye can we see it. We can see it. See here, when light passes through the cell, small phase shift occur which are invisible to the human eye. In phase contrast microscopy, these phase shift are converted into the change in the amplitude. This amplitude, this amplitude, this amplitude, which can be observed as a difference in a image contrast. Thank you for listening me, and this is a reference link, from that I made this PPT, and thank you everyone, and thank you ma'am for listening me. Have a nice day. Bye.